Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we are just going through the acute inflammatory response at the moment, just so that the adaptive immune response sits in place alongside the acute inflammatory response, or at least following the acute inflammatory response. Okay, right. So far what we've seen is that in response to a pathogen infecting a portion of our tissue, what happens is that the sentinel cells, which include the dendritic cells, the resident macrophages, and the mast cells, these become activated and they release pro-inflammatory mediators. And resident macrophages and dendritic cells release the pro-inflammatory cytokines, interleukin-1 and tumor necrosis factor alpha, whilst the mast cells degranulate and release histamine. These pro-inflammatory cytokines and histamine all act on endothelial cells of the blood vessels that are within that portion of tissue, so the arterioles, the capillaries, and the venules. Okay, and we're at the moment discussing what histamine is going to cause, because histamine causes what's known as type 1 activation of endothelial cells, which occurs within minutes, whereas interleukin-1 and tumor necrosis factor alpha, they cause what we'll come on to, which is known as type 2 activation, which takes hours to initiate because it involves changes in gene expression. Okay, right. So we've discussed that histamine in type 1 activation causes the release of vasodilatory molecules, which are going to have an effect within the arterioles and cause relaxation of the smooth muscle cells, and that will lead to the vasodilatation of those arterioles, which will lead to increased blood flow to the area, which will make the area red and hot to the touch. Okay, and the purpose of this is to bring in more blood to the affected area so that we can bring out more troops from the blood to come and attack this pathogen. Okay, so how do we actually bring the troops out of the blood? Well, one of the ways is that we cause endothelial contraction. Okay, so type 1 activation also leads to contraction of the endothelial cells, which opens up these gaps between the endothelial cells by pulling apart the tight junctions and the adherens junctions. Okay, right. So that allows fluid from the blood to move out of the blood through this gap into the interstitial fluid, and within this inflammatory exudate, as it's called, which is the name for this fluid leaving the bloodstream and going into the interstitial fluid, there will be complement proteins and coagulation factors. Okay, so let me get rid of that S there. Okay, right. And the complement proteins can set up cascades when they actually attack the cells directly, the pathogen, whether it's a cell in the case of fungi and bacteria and parasites, or whether it's a virion particle in the case of viruses. And the coagulation factors, they set off the coagulation cascades when they come into the interstitial fluid, and that causes the production of loads of fibrin strands, which form a fibrin meshwork that encapsulates everything and sort of binds the pathogen in, basically, and stops it from spreading. Okay, now the next portion of type 1 activation of endothelial cells is going to be that the endothelial cells will now start to recruit leukocytes out from the bloodstream, and they'll move them into the interstitial fluid. So we're going to remove white blood cells, and the fancy word for white blood cells is to call them leukocytes. We're going to move them out of the bloodstream and into the interstitial fluid. Now, the leukocytes which are going to be recruited in the acute inflammatory response at first are going to be neutrophils. Okay, so type 1 activation leads to the extravasation of neutrophils. Okay, and extravasation just means moving neutrophils from the vascular system into the interstitial fluid. Okay, so let's just talk about neutrophils for a bit. So neutrophils are a phagocyte, so they can engulf pathogens and destroy them, basically, break them down. And if we were to draw a picture of a neutrophil, we draw something that looks like this. The nucleus is said to be multi-lobed, okay? So the nucleus doesn't have a conventional structure. Instead it has these multiple lobes, like so, okay? And the number of lobes that a neutrophil nucleus has is not set. Okay, so different neutrophils will have differing numbers of lobes within their nucleus. And this has led to neutrophils also being known as polymorphonuclear cells. So poly means many, morpho means shape, nuclear refers to the nucleus, so many shaped nucleus cells. Okay, so polymorphonuclear cells. Okay, and they can also be referred to as polymorphonucleocytes, which is final name for them. So this is really combining the nuclear and the cells together to make polymorphonucleocytes. So site means cell. Okay, and for this reason you can see neutrophils often referred to as PMNs for short, which stands for polymorphonucleocytes. Okay, finally you can also just hear them referred to as polymorphs. So they have lots of different names, but it's all referring to the same cell type. Okay, so basically in response to histamine, the endothelial cells are going to start becoming sticky for neutrophils, and then they're going to grab neutrophils from the bloodstream, and then they're going to help them to cross between the gaps in the endothelial cells. So if we have an endothelial cell here, okay, and its neighbouring endothelial cell is here, then we've already discussed that they've started to open up a little gap due to endothelial contraction. Okay, and now what's going to happen is that they're going to help this neutrophil to sliver through this gap here between the two endothelial cells. Okay, so here's the nucleus of the polymorphonucleocyte, or the neutrophil. Okay, and this process of the neutrophil slithering through the gap between the endothelial cells is known as diapedesis. Okay, and this allows the neutrophil to leave the bloodstream and go into the interstitial fluid. And once it's there, what it will do is it will phagocytose pathogens. So it will engulf them into a phagosome and then fuse lysosomes with the phagosome in order to break down the pathogens. So what will happen is, if we draw this phagocytosis process here, when it finds a microbe, what will happen is it will invaginate its membrane like so. So here's a microbe here. So here's the nucleus again, this this multi-lobed nucleus structure. Okay, and I have to shade it in black now, can't resist. Okay, right, so let me colour in the microbe in red. So what will happen is that initially you'll invaginate the membrane. Well, actually initially the microbe will bind to proteins on the surface of the neutrophil, and then this will cause the invagination of the membrane around that parasite, and then what will happen is this invagination will pinch off, and you'll end up with a vesicle inside the cytoplasm of the cell with that pathogen inside. Okay, so there it is. And this sort of a vesicle here inside the cytoplasm of a neutrophil is what's known as a phagosome. Okay, and then what will happen is that you'll have other little vesicles here, which are known as lysosomes. These are lysosomes. And these lysosomes are full of enzymes known as lysozymes. Okay, so let me just move this up a little bit. And basically what will happen is these lysosomes will move towards the phagosome fuse with the phagosome and release their lysozymes into the phagosome, and then the lysozymes will break down the pathogen, okay? So this process of engulfing a pathogen and then breaking it down is a process known as phagocytosis, okay? And neutrophils are an example of a phagocyte. They're kind of the cheaper version of a phagocyte, okay? So I often like to compare the members of the immune system to chess, and neutrophils are kind of like the pawns of the immune system. They're something that you have a lot of and you don't really care whether or not they die, okay? But this is what they are. They are a phagocyte. They will go in, the body sends them in first as part of the innate immune system, and they will go in and you'll pile them in there and they'll just engulf things and destroy them, hopefully. So, they're what's known as a phagocyte, and neutrophils are an example of a phagocyte, and we're going to see the other example of a phagocyte is a macrophage, and we'll discuss this more in a moment. Okay, so macrophages are the other examples of phagocytes, the other archetypal example of phagocytes. Okay then, so type 1 activation of endothelial cells due to histamine then, what you get is vasodilatation of the arterioles leading to the affected area. And this means you get a greater blood flow there, which allows you to bring in more leukocytes and more proteins as well. Okay, endothelial cell contraction allows the formation of an inflammatory exudate, which contains the complement proteins and the coagulation factors, which are both involved in attacking and containing the pathogen. Okay, and finally you get neutrophil extravasation. So neutrophils stick to the surface of the endothelial cells and will be moved through the gaps between the endothelial cells, and then will go into the interstitial fluid and phagocytose pathogens. Okay, right. So that's what histamine does to the endothelial cells. Interleukin-1 and tumor necrosis factor alpha cause a different type, but very much so related type, of activation in endothelial cells. So these, remember, were both released by dendritic cells and resident macrophages when they got the alarm signal that a pathogen was present. And these are going to cause what's known as type 2 activation of the endothelial cells. Okay, now type 2 activation is activated much slower than type 1 activation. Type 1 activation will occur in minutes. Type 2 activation takes hours, and the reason type 2 activation takes so long is because it involves an actual change in gene expression within these endothelial cells. Okay, so the changes in gene expression that occur within type 2 activation are going to lead to actions that will help what has already started with type 1 activation. So type 1 activation, everything you did was already there. You just activated a bunch of molecules that were already there, which is why it was so quick. Type 2 activation, you actually change which molecules are there, basically. Now, the actual things that you do, however, are just helping what you've already started with in type 1 activation. So again, you release vasorelaxant molecules. So type 2 activation is going to increase the amount of vasorelaxant molecules you're producing hugely. Okay, so it's going to cause vasodilatation of the arterioles, basically. So the endothelial cells in the arterioles are going to undergo type 2 activation in response to interleukin-1 and TNF-alpha, and they're going to start releasing even more vasorelaxant molecules than they were in response to histamine and type 1 activation. Okay, so upregulates the vasorelaxant molecule secretion and therefore increases vasodilatation and therefore increases blood supply even more. Okay, it also causes increased leakiness of the endothelial cells. So the fancy word for increased leakiness is to say increased vascular permeability. Now, it's going to do this through a different mechanism than endothelial cell contraction. So in endothelial cell contraction, remember, what we did was pull the tight and adherens junctions apart. In type 2 activation, what you're actually going to do is dismantle the cells. So let me talk you through this. So, if we've got our two neighbouring endothelial cells here, then what is actually, we need to ask a question basically. What is actually holding up the cell? Why does the cell remain in the shape that it does? Well, basically it's because the cell has a very dense skeleton known as the cytoskeleton. So let me write this word down because it's so important. So the cell has what's known as a cytoskeleton. So remember, cyto means cell, and then skeleton means support structure. Okay, so inside a cell, it's absolutely full of an intertwining spider's meshwork, basically, of cytoskeletal proteins, and there are two major filaments that you have. You have actin filaments, which are made up of polymers, of actin monomers, and you also have tubulin filaments, okay, which are made up of polymers of tubulin. Okay, and these are two of the major cytoskeletal components. Okay, now if you were to dismantle all of the actin and tubulin filaments in this edge of the endothelial cell over here, then what would happen is the cell membrane would just flop. It wouldn't maintain its structure if it wasn't being held up by the cytoskeleton, so it would flop back. So that's exactly what you're going to do. You're going to dismantle the actin and tubulin cytoskeletal components in the edges of these endothelial cells. So in these bits that I'm shading in turquoise, you are going to dismantle the actin and tubulin cytoskeletons, okay? And after you've done that, the cell membrane will just flop down, basically. So what you can see is that endothelial cells are going to retract basically backwards, and they'll open up much larger gaps between the two endothelial cells. So this gives a more permanent hole between the endothelial cells than did endothelial contraction as a result of type 1 activation. Okay, so again, this will allow even more fluid to leave the capillaries and the post-capillary venules, which are in your tissue, and this will lead to a bigger inflammatory exudate, more complement proteins, more coagulation factors, more innate immune system ready to attack the pathogens, basically. So that's what how we achieve increased vascular permeability in type 2 activation. Finally, it's also going to help with leukocyte recruitment. And this is where type 2 activation changes over time, okay? So initially, when you first activate type 2 activation of endothelial cells, then it will increase neutrophil extravasation. So it will increase neutrophil recruitment. So initially, type 2 activation will continue the work of type 1 activation. Okay, so it will help to recruit neutrophils, and you'll recruit loads and loads more neutrophils into the interstitial space. Okay, so it puts proteins on the surface of the endothelial cells that help grab onto neutrophils and then move them through these now bigger gaps between the endothelial cells. Okay, but then after around 6 to 24 hours, so where should I jot this down? Over here, just because there's no space over there. So after around 6 to 24 hours, what happens is the type 2 activation changes from promoting neutrophil extravasation to promoting monocyte extravasation. Okay, so let me discuss monocytes with you. So I'll get another piece of paper. Okay, so a monocyte is a type of cell that circulates within the bloodstream, the structure of which looks kind of like this. Okay, so it has a single nucleus like this. So this is a monocyte, and it is circulating within the bloodstream. Now, after around 6 to 24 hours, type 2 activated endothelial cells will start expressing molecules on their surface which are going to grab onto monocytes, okay? And then the monocytes will diapodese, through the spaces between the endothelial cells and get into the interstitial fluids. You're going to get monocyte recruitment in the same way as you get neutrophil recruitment. Okay, so after a while, basically, the endothelial cells stop recruiting neutrophils and start recruiting monocytes instead. Now, when monocytes get into the interstitial fluid, basically they differentiate into a different type of cell. Okay, so they differentiate now into what's known as a macrophage. So it's important that you understand that a monocyte is not the same as a macrophage. A monocyte is a precursor to a macrophage, and the structure of the cell does change when it moves into the interstitial fluid. So this process is the process of differentiation. Okay, so the cell has actually changed type, basically. Okay, and now macrophages are another phagocyte, basically, but they're a slightly more powerful phagocyte than neutrophils. Neutrophils are weak. They're inexpensive cells that you have in a huge number, and therefore you just throw them out. If neutrophils are pawns, macrophages might be a rook or something. They're a little bit more powerful, a little bit more expensive, a little bit more powerful, and they're capable of phagocytosing pathogens, basically. Okay, so that now concludes our discussion of the acute inflammatory response. So, basically what happens is the sentinel cells cause type 1 and type 2 activation of endothelial cells, and these lead to vasodilatation of the arterioles, which lead to the affected area, which increases blood flow to that area, causing redness and also heat at the affected area, rubor and calor. Okay, and then what you also get is increased vascular permeability in the capillaries and post-capillary venules, which leads to fluid moving out of the bloodstream into the interstitial space, and this causes a swelling, and this is known as an inflammatory exudate. Now, the purpose of this inflammatory exudate is to bring all sorts of proteins that are usually in inactive forms in the blood out into the interstitial fluid. Two notable examples are the complement proteins and the coagulation factors. The complement proteins will directly attack pathogens. The coagulation factors will undergo both the intrinsic and extrinsic coagulation cascades when they arrive in the interstitial fluid, because they will be exposed to collagen and tissue factor, which are in the interstitial fluid, and those trigger off the intrinsic and extrinsic coagulation cascade respectively. And this will lead to the conversion of fibrinogen into fibrin, and then fibrin will assemble into polymers and form huge great strands which intertwine and form a very dense meshwork known as a fibrin meshwork, which will enclose the pathogen and hopefully contain it, basically stop it from spreading. Okay, then finally what you get is leukocyte recruitment. So initially you recruit neutrophils in huge numbers, which will come in and start phagocytosing the invading pathogen, and then you change to starting to recruit monocytes, which when they move into the interstitial space differentiate into macrophages, which are also going to phagocytose the invading pathogen. In the next video, what we will do is begin to discuss now the adaptive immune response.